For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
Okay, looks like we are live tonight. We have an awesome debate for you guys. I'm really pumped. Tonight we're debating intelligent design versus naturalistic evolution. We've got Otangelo Grasso here taking on David Neff. Both of these gentlemen are no stranger to debates, nor are they stranger to this channel. So they both have had many debates, and they've been studying this topic for years, from my understanding. So this is definitely going to be one to remember. Before we introduce the debaters, though, I want to thank the audience. It's good to see everybody here. We've already got some super chats flying in, so let me read those real quick. We've got Doki Doki Bible Club, $35 super chat. Doki, thank you so much for all the support. You guys are awesome. You guys are the reasons why we're able to do all these streams, host these debates, interviews, discussions. So these are important topics, and we love to do as many streams as possible, of course. So Doki Doki says Sal is doing an after show. Come teach him how to work StreamYard. So Salvador Cordova is going to be having an after show on his channel. So make sure you guys are there for it. I'll announce it throughout the debate too for when we get new people in. Doki Doki, super sticker, thank you so much. Also, yes, there's going to be a question and answer. So make sure you are tagging me with your questions, and I'll save as many as I can, and we will get to those at the end of the debate. So another super chat here. Brother George Bond says, love you all. God bless. God bless you as well. Thank you guys for all the support. And super chat from Doki Doki says, for David, please double major in science so I can call you a musician scientist.
Well, I'm actually lucky for you. That is actually what I've been planning on doing.
Well, there you go. There you go. And George Bond says, piano music, please. George, the second I seen you demand the piano music, I put it on just for you.
Hey, you know what would be actually kind of fun? Standing, if you can find what pieces they are, I could learn how to play them on the piano and post a video of me doing that for you guys.
There you go. There you go. We'll make you world famous. So, anyways, let's. I'm going to go over the structure real quick. So we're going to be doing 12-minute openings. We're going to be starting with Otangelo, followed by. Are we doing a 6-minute rebuttal, gentlemen? Remind me what.
That sounds fun. That sounds fine. 6-minute rebuttal. So
6-minute uninterrupted rebuttal. Then we're going to go right into an open discussion. Both of these gentlemen are respectful, cordial. I'm sure this will be extremely easy to moderate. Otangelo, I saw you in the chat. I'll make you a moderator so you can post the links, brother. So I'll do that right when we're done here. And then we'll do we'll wrap things up with concluding statements, and then we'll do a Q&A. So enough from me. We want to get to the debate, and we want to get to the guests tonight. So I'm going to hand it over. Before we do an opening statement, though, I want to just do a brief introduction, guys. Break the ice. Tell us a little bit about yourselves. Tell us what you've been doing over at your channel. You can plug your channels if you'd like to. So let's start off with David. Thanks for giving us your time for tonight's debate. How you doing?
Hey, I'm doing quite well. And just to clarify, is there going to be kind of like an open discussion between me and Otangelo after the rebuttals?
Oh, yes. Yep. Okay. After the rebuttal, we're going to go into open discussion.
All right, sure. So my name is David Neff. As Doki pretty much already called me out, I am a musician, and I am passionate about science and music. I play piano, and I also dabble a little bit on the organ. I actually just posted a couple of videos of me playing the organ last week, so you should check them out on my channel. I am also doing a stream this week on homeopathy and debunking that. So there's something to look forward to. So go over to my channel, hit the subscribe button. And one more thing, I am still working on the Foundational Fallacies of Creationism series. So definitely hit the subscribe button and be notified when that comes out.
Awesome. Well, thank you so much for that, David. Just from the chat, looked like a couple people said there was some fuzzy mic going on. Otangelo, I did mute you just while David was giving his intro. I think it might be coming from your end. If we can fix it, whether maybe right now you're using your mic, so maybe whatever we can do to fix it. But let me unmute you real quick so we can get your introduction. And Otangelo, thank you so much for being here. Actually, it looks like the background noise is gone, so I think we might have solved the problem. Otangelo, a little bit about yourself. Thanks for giving us your time. Thanks for being here.
Okay, it's great to be at your channel, Standing for Truth. I think most on the audience, they know me already. I am regularly debating on YouTube about these topics. This is the first time actually that I am debating about evolution. I've never done it before, so it will be my first time. It will be great. And I have 2 channels on YouTube. One is The God Talk, which is focused to have chats. And the other is Intelligent Design Academy, where I upload videos in regards of intelligent design. And I have also a virtual library. It's reasonandscience.catsport.com, where I have a virtual library and put all my articles so they are public. Everyone can read about these topics which I study and am passionate about.
Awesome. Well, thank you so much, Otangelo. What I'll do is add your website and your channel into the links into the description box. Anything that you guys want me to add to the description box after the debate, let me know and I'll put that in there. So yeah, looks good. Everybody's saying the audio sounds good. So I'm glad we got that out of the way. Everything is fixed. Thank you, gentlemen, again, for giving us your time. Great intros. So we are pumped. Guys, we got 30 people in the chat. So start gathering questions, and of course tag me and I'll save them, and we'll get right into it. So Otangelo, you're going to be starting off with your opening statement. We can get your screen shared if you would like to, and then I will set the timer whenever you're ready, of course.
Yeah,
and
I'm ready.
Then let's see, and we'll add that to the stream. Boom, looks like it's good to go. And everybody in the chat saying everything is good. So why don't we get it started? Whenever you're ready, Otangelo, just give me the A-OK and I'll start the timer.
Yeah, you can start. Okay, intelligent design against naturalistic evolution. What is the better explanation of origins? There are 4 facts which all agree upon in regards of evolution. And there are 2 which are disputed between the proponents of intelligent design and the proponents of evolution, namely common ancestry and the origin of biodiversity and higher organismal form. In regards of common ancestry, there are 6 major points which evidence that the claim is not true. The major transitions in evolution are divided in 6 major steps. In the evolutionary narrative, at the root of the evolutionary tree, there is the split of the 3 domains of life. The origin of eukaryotic cells is acknowledged to be a major evolutionary problem. Eukaryotic cells function on different physical principles compared to prokaryotic cells, which is directly due to their size, which is up to 1,000 times larger in diameter and having a volume size up to 15,000 times larger. The diversity of the outcomes of phylogenetic analysis has led to considerable frustration and phylogenomic impasse owing to the inadequacy of the available phylogenetic methods to remove the ambiguity of relationships. Still today, most biology textbooks tell us that organelles such as chloroplasts and mitochondria evolved through endosymbiosis, a hypothesis promoted by Lynn Margulis, and that is supposed to explain the origin of eukaryotes. Because mitochondria look like prokaryotes, it was assumed that eukaryotic cells came into existence when one prokaryotic swallowed another prokaryote. A problem with this idea is that prokaryotic cells lack the ability to swallow other cells. Eukaryotic cells can, but every eukaryote that we know has mitochondria. It's not clear which came first, the ability to swallow other cells or the mitochondria. Then in 2015, some science papers announced that the link between prokaryotes and eukaryotes had been found. And then in Nature magazine, they published that modern phylogenetics indicates that the host, which engulfed another bacteria to become mitochondria, was an archaeon. Only 1 year later, they had to acknowledge that there was no relatedness and that the results indicated that Lochiarchaea did not correspond to Eukarya. There is a considerable number of innovations that would have had to take place to transition and evolve from prokaryotic to eukaryotic cells. I am listing over 40. One of the major problems is the origin of organelles in eukaryotic cells. The Golgi apparatus, the nucleus, the mitochondrion, etc., require information in the organelle from the mother cell to make organelles for the next generation, its daughter cells. That raises the question how these organelles evolved if the first could not inherit the information from the mother cell. Daughter cell membranes come only from mother cell membranes. When a cell reproduces by division, it has to duplicate its organelles in addition to its chromosomes. In general, cells do this by incorporating new molecules into the existing organelles, thereby enlarging them, and the enlarged organelles then divide and are distributed to the daughter cells. Thus, each daughter cell inherits a complete set of specialized cell membranes from its mother. During the process, the organelles are enlarged and subsequently must divide. But how does this happen? We all know how a boa kills its prey, by constriction. Amazingly, there is a set of proteins who perform their task like a boa constriction. They are called dynamine and operate by an amazing process called organelle constriction. They possess the remarkable property of assembling into contractile helical polymers that wrap around the neck of an organelle. They perform the challenging job of constricting the organelles until they are separated. This is just one example of many highly orchestrated and finely adjusted processes that have to take place during organelle constriction. In my understanding, this process had to emerge fully functional right from the beginning. A stepwise evolutionary process seems not feasible. To me, this looks like amazing molecular engineering and design. Another huge problem is the origin of mitotic cell division, which is one of the most fundamental tasks of eukaryotic cells. Cells have intricate and tightly regulated machinery to ensure that mitosis occurs with appropriate frequency and high fidelity. At least 14 cell cycle regulators are essential. If one of those regulators is missing, the cell cycle cannot be completed. Therefore, the regulation of the cell cycle is irreducibly complex. To my next point, 5 geochemical energy cycles are essential for advanced life forms on Earth. That creates a huge problem for evolutionary scenarios. How did these cycles get off the hook? These are gigantic interdependent systems where if one part of the cycle is missing, nothing goes. And they are also interdependent between each other. The 3 main cycles are the water cycle, the carbon cycle, and the nitrogen cycle. These 3 cycles, working in balance, are responsible replenishing the ecosystem with the nutrients necessary to sustain life. If any of these 3 cycles should become unbalanced, the effects on the ecosystem are catastrophic. Without cyanobacteria, not enough fixed nitrogen would be available. Without fixed nitrogen, no DNA, no amino acids, no proteins can be synthesized, and no life would probably exist. This is an interdependent system. It cannot have evolved in small steps. All must exist at once. In the evolutionary narrative, life started in an atmosphere that was anoxic, and only anaerobic life forms existed. It is supposed that life started about 4 billion years ago, and about 2.5 billion years ago, oxygen started to fill the atmosphere through cyanobacteria, performing photosynthesis, and by this, they produced their food, glucose. Oxygenic photosynthesis and nitrogen fixation are conflicting metabolic systems. Nitrogenase is the enzyme that fixes nitrogen, but is oxygen sensitive. As soon as it is exposed to oxygen, the enzymatic reaction shuts down, and oxygen produces as well hydroxyl radicals, which wrecks DNA. So as soon as cyanobacteria would have started to produce oxygen, they would have killed themselves. So why would cyanobacteria have evolved protective enzymes if the need for them didn't even exist yet? Nick Lane describes the dilemma in the book Oxygen, the Molecule That Made the World. Before cells could commit to oxygenic photosynthesis, they must have learned to deal with its toxic waste, or they would surely have been killed. But how could they adapt to oxygen if they were not yet producing it? The origin of oxygenic photosynthesis very early in the evolutionary timeline is another huge unsolved problem. There would be no advanced life forms on Earth without this pathway, which is almost the most complex biosynthesis process known. In photosynthesis, 26 protein complexes and enzymes are required. A good part of the protein complexes is uniquely used in photosynthesis. The pathway must go all the way through, and all steps are required, otherwise glucose, which is what cyanobacteria consume for food, is not produced. The supposed evolution of multicellular life forms from simpler unicellular microbes is another pivotal moment in the history of biology on Earth. Biologists can provide plenty of guesses about how multicellular organisms could have evolved from unicellular organisms. Today, most plants, animals, and fungi are composed of multiple cells that work together as a single being. The fact is that science is basically in the dark of how this transition actually happened. And there is a good reason for this. Explaining organismal architecture depends on explaining how organs, tissues, and cells form and gain shape. On the lowest level, there are at least 19 different specifications required for each single cell. So now to the last and most important question. If unguided evolutionary mechanisms do not explain the origin of organismal form, what does? The real mechanisms to explain organismal complexity are prescriptive, pre-programmed, instructional, complex information encoded through various genetic codes and about 30 epigenetic codes, signaling and communication using various signaling networks that act on a systems level and are all involved in organismal development, responding to environmental cues and nutrition demands. Here, I am listing 19 different mechanisms which operate on a systems level. That brings us, without any doubt, to an intelligent designer. Thank you.
Awesome. Just on time there, Otangelo. 12 minutes right on the dot. So, David, we're going to be handing it over to you real quick, though. So it looks like we've got some good questions coming in.
Awesome.
Guys, good job. Make sure you're tagging me. And let me reset the timer. David, let me share your screen for you. And I will start your timer whenever you're ready. You just give me the cue, and I'll start
Okay.
the timer.
Okay, one moment. All right, you can start the timer. Well, once again, thank you, Standing for Truth, for hosting this debate, and Otangelo for agreeing to this debate. Note to self, never do a debate after a 10-hour shift ever again. I am falling asleep, but thankfully I have some caffeine to keep me awake. So I kind of want to take this debate from a little bit of a different angle. And instead of just, I'm going to kind of break this down into three main parts. Number one, I'm just going briefly over the evidence for evolution, but more importantly, in part two, I really wanted to kind of go into how evolution is actually used and the practical applications of it. And then finally, I am going to discuss some of the many problems associated with intelligent design. So nothing in biology makes sense except in the light of evolution. Evolution is the cornerstone of modern biology. Nothing would make sense without this theory. And Dr. Daniel Fairbanks said that having applied evolution theory in my research, I can unhesitantly say that evolution never mattered more than it does now. Using the theory of evolution, we have been able to learn so much about the development of life and have been able to learn so much about medicine and ecology and all of that fun stuff. First point is evolution is an observable fact. The microevolution that he and I both agree happens, and we both agree speciation happens, and macroevolution as well. So both speciation is macroevolution. So Otangelo, Standing for Truth, and myself all agree with macroevolution, both microevolution and macroevolution. The points of disagreement are the time scale, what role God may have played, and the extent of common ancestry. This debate really is going to be homing in on the final point of the disagreement. Does all of life share a common ancestor, or do only certain organisms share a common ancestor? So there are, because we know several different modes of speciation, and we even understand how this happens very well. We even have different names for them and how they happen. And an analogy for this speciation is language. Language evolves over time the same way biological organisms do, descent with basic inherited modification. I inherit the language from my parents, but we also, over time, add new words, and words change meaning, and over time, we get new languages. And it's the exact same way in biology. So point number one is evolution helps us understand the development of life. Here is a very good paper about the developments, the speciation of cats. The paper says the ability to hybridize is the most important criteria for including species within a common basic type. This criterion cannot be used directly on fossil forms, however, because it indicates the extent of morphogenetic potential for a basic type. Hybridization is an indirect indicator of fossil inclusion. Therefore, reports of extant cat hybridization, fossils, skeletal evidence, and various other features, including molecular sequencing data, etc. Felidae represents a clear delineated basic types. This is ironically from Creation Ministries International. What they actually did was they took this image from an actual peer-reviewed paper, which I'll show you in just a minute. And what they did was just remove the timescale that doesn't agree with their point of view. And I think that is very dishonest on the part of Creation Ministry International. Here's the original picture. And here's the actual abstract from that paper. Oh, I think I it out real quick. There we go. Modern field species descended from relatively recent 11 million divergence, and this is from the genetic analysis of that. And using the molecular timescale and the molecular clock, we can guesstimate when these divergence happened. Now let's look at what role evolution has in our health. Number one, evolution plays a back and forth game between the pathogens and us. As we confer resistance to the antibiotics, as we evolve resistance to the pathogen becomes stronger to overcome us. And the evolutionary framework helps us to identify the origins of novel viruses. Evolution explains why pathogens counter our attempts to control them, and Standing for Truth, being a nurse, knows this quite well. And this is quite a huge problem with the antibiotic-resistant bacteria. That is a huge problem in medicine today. And finally, evolutionary genomics advances medicine. So let's look at HIV, which let's look at the actual steps that was needed in order for it to actually infect humans. First is that, excuse me, let me go back to my notes here. So from Dr. Fairbanks' book, the virus had to mutate into a form that can overcome natural immunity to SIV in humans. Mammals have a gene that encodes a protein called tetherin. This protein has evolved to confer resistance to retroviruses by tethering them to the inside of the cell they infect and preventing the virus from replicating. For SIV-CPZ to successfully infect a human, it had to overcome the suppressive effect of human tethering. The SIV-CPZ evolved by acquiring two anti-tetherin genes called NEF and VPU, one from each of the original monkey viruses that fused to form the chimpanzee virus. The NEF gene mutated to overcome the chimpanzee tetherin, but the VPU gene remained essentially insert. When the virus jumped to humans, the human tetherin was so different that the NEF gene could not overcome human tetherin. Instead, the VPU gene mutated to overcome human tetherin, allowing HIV-1 group M to infect humans. And a mutation in the second gene called GAG was also required for the chimpanzee virus to jump to humans. So by all metrics, let me get back to where I was. By all metrics, this is an irreducible complex virus, which seems to be Otangelo's entire opening statement was basically irreducible complexity. Well, here is a virus that is irreducibly complex. Many mutations and parts had to come together exactly correct, and we know that this is exactly the pathway for which this virus evolved to infect humans. And finally, my point against intelligent design is this: intelligent design is unintelligently designed. What does the word God even mean? Who or what is the designer? What is their nature? What is the mechanism by which the intelligent designer created? What is the limit to common ancestry? And in my rebuttal, I will be honing on this point quite forcefully. And when did this creation happen? And which God did it? Was it the Christian God, the Jewish God, the Muslim God, a deist God, any God? If intelligent design advocates want to replace the theory of evolution, they need to answer these questions and give an objectionable evidence for it. So again, I am going to be pressing Otangelo to answer each of these questions in the open discussion. Because if he can't even answer these basic questions, then intelligent design, I'm sorry to say, cannot be science. So in summary, evolutionary principles help us to know who we are and where we came from. Evolution has practical applications. Intelligent design simply does not. And intelligent design is poorly designed. Thank you very much. How did I do on time?
Awesome. Both of you, actually. I had to, boom. There's still, you still got 4 minutes. So what I can do is toss those 4 minutes into the discussion. Typically is people's favorite part of the debates anyways. So both of you did great on time. Next phase is going to be the rebuttal round. But real quick, I forgot to do this in the announcement. So guys, be here this Saturday, 9 p.m. sharp. We're going to have Derek Barnes back on here debating the film free on the flood. So another good debate to look forward to. So guys, thanks for your opening presentations. Clear, concise. Thanks so much for the visuals. I know it takes work to prepare those. So good job. We're going to give it over to Otangelo. You've got 6 minutes uninterrupted rebuttal time. I'll give a 1 minute warning when we reach the 5 minute mark, Otangelo, but whenever you're ready on your first word, I'll start the timer.
Okay, I made some notes here, and I would like to use again the to show my replies. Okay, so
of course, yeah, I will. I'm not going to start your timer just yet. Once we get your screen shared is when I'll start it, to be fair, Otangelo. So once you hit screen share,
yeah, I'm just hitting it.
There we go.
Okay.
I got it. I added it to the screen. You're good to.
Let me put it bigger so people can actually follow what I'm writing here. Okay, I think that David he touched again in regards of common ancestry. And here I am lining out common descent, the tree of life, a failed hypothesis. I didn't have time in my introduction to expose the main points which demonstrate that common ancestry is not a plausible explanation for how everything started. And here, these are the main points. Point one, the DNA replication machinery is not homologous in the three domains of life. Bacteria and archaea differ strikingly in the chemistry of their membrane lipids. Three, sequences of glycolytic enzymes differ between archaea and bacteria and eukaryotes. Then four, there are at least 6 distinct autotrophic carbon fixation pathways. And if common ancestry would be true, there should be just the Wood-Ljungdahl pathway and that dominating the entire spectrum of life forms. But we have 6 different carbon fixation systems. And the big question is, why is that so? And how could they have evolved into 6 different ones? And even more, like the photosynthesis pathway, the fixation of carbon there is in an aerobic environment. And that would require a transition from anaerobic carbon fixation to aerobic carbon fixation. That's a huge problem because the enzymes that are operating in an anoxygenic environment, they would be shut up if they encounter oxygen. So how that transition could have occurred is a big enigma. Then 6, the typical eukaryotic cell, as I said in my introduction, is about 1,000 times bigger by volume than the typical bacterium. So how that transition from a less universal common ancestor to eukaryotes could have occurred, that's a big question mark. And there are 7, 8, and 9 points, but I will not get closer into this. Then in macroevolution, there has to be made a distinction between primary speciation and secondary speciation. Secondary speciation is what we can actually observe, which is the separation of populations, which then after some time cannot interbreed anymore. That's macroevolution on a secondary speciation level. But on a primary speciation level, which is the big question mark, that has never been observed, which is actually the emergence of big organismal form and changes and evolution of new limbs like arms, like eyes, like ears, and limbs, and so forth. That is the big issue here. And just to give an example, let's suppose that you have a snake and that has to grow legs or to become to have 4 legs and to become a tetrapod. So what would have to change in order to have the evolution of a new limb? And I am exposing here 7 different systems which work in an interdependent manner and joint venture together. And all of those had to evolve simultaneously together. That would be first the muscular system, then secondly the skeletal system, the nervous system, 4th the endocrine system, 5th the circulatory system, 6th the integumentary system, and 7th the lymphatic system. And this is, in my understanding, an irreducible system which has to evolve all together, or it cannot evolve. If you just evolve the muscular system, then the rest is missing. You can't evolve a limb. And if you have just 6 of these 7 points made here, then you can't have the evolution of a new member of an organism. So this is a big problem in my understanding. Then cats, which David has shown. Cats, of course, is microevolution. That is a no issue. Then regards of viruses, I would say yes, that is also microevolution. That's not a big issue. Then David was pointing out about how we can recognize signature of intelligent actions. That is actually a very good question. I have studied this, how we can make a distinction about things and features which are only the signature and imprint of intelligence, which unguided random events cannot do. And that is actually a very important and good distinction and demonstration why intelligent design tops natural evolutionary explanations to explain the origins of biodiversity. And I am pointing out 9 different main points which demonstrate
You got 1
things.
minute, Otangelo, 1 minute.
Okay, which demonstrates things that intelligence can do and non-intelligence can't. And these are all things that we can observe in nature. Point 1, implementing things based on the regular behavior, order, mathematical rules, laws, principles, and physical constants and logic gates. 2, something purposefully and intentionally developed and made to accomplish a specific goal. 3, repeating a variety of complex actions with precision based on methods that obey instructions governed by rules. 4, an instructional complex blueprint or protocol to make objects like machines, factories, houses, etc., which are irreducibly complex, integrated, and interdependent.
10 seconds.
And 5, artifacts which use might be employed in different systems. 6, things that are precisely adjusted and finely tuned to perform specific functions and purposes. Okay, I will stop here.
Awesome. Just on time. These openings and rebuttals are flying by, I'm telling you. Thanks again to people in the chat tagging me with your questions. So we are now moving over to David. David, it is your 6-minute rebuttal. Whenever you're ready, let me know and I'll start the timer.
All right. Yeah, I think I am ready. Thank you, Otangelo, for that incredible rebuttal, and thank you once again for that fantastic opening presentation. What I kind of want to do is that we both agree that speciation happens at least on some level. What I would really like to do in the open discussion is kind of look at some of the cladistics and try and see if we can find out which clades are related and which clades are not. That way, I think this will be important to do in the open discussion, which I'll explain why in a little bit. But let's go into the issue of irreducible complexity, since your entire arguments, almost your entire argument is based on irreducible complexity. In 2005, there was a huge court case over teaching intelligent design in public school. Professor Michael Behe was brought up irreducible complexity in the court of law. And the court, the judge, who was a Bush-appointed judge, stated pretty clearly, Professor Behe's claim for irreducible complexity has been refuted in peer-reviewed research papers and been rejected by the scientific community at large. And the reason for that is, and this is why, I mean, we see irreducible complex organisms that we know how they evolved, such as the HIV virus that I presented in my opening case. And I think Otangelo would agree that HIV did in fact evolve from SIV, and it is by all metrics irreducible complex. So I don't see irreducible complexity as any challenge for evolution at all. But I kind of wanted to take a little bit of a look at what the Dover trial, what the trial actually found. Because there's a lot of really good information in here for it. After a searching review of records in applicable case law, we find that ID arguments may be a true proposition with which the court takes no position. ID is not science. We find that ID fails on 3 different levels, any one of which is sufficient to preclude any determination that ID is science. They are, number 1, that it violates the centuries-old ground rules of science by invoking and permitting supernatural causation. And 2, the most importantly, the argument of irreducible complexity, central to ID and central to Otangelo's case, employs the same flawed and illogical contrived dualism that doomed creation since the 1980s, and ID's negative attacks on evolution has been refuted by the scientific community. As we will discuss in more detail below, it is additionally important to note that ID has failed to gain acceptance in the scientific community. It has not generated peer-reviewed publications, nor has it been the subject of testing and research. So further on in this court of law. As referenced, the concept of irreducible complexity is a negative argument against evolution, not proof of design, a point conceded by the experts, Professor Minch. Irreducible complexity, quote, is not a test of intelligent design, it is a test of evolution. Irreducible complexity additionally fails to make a positive science case for intelligent design, as will be elaborated upon below. We note that the irreducible complexity as defined by Professor Behe and Darwin's black box and subsequently modified article appears as follows. By irreducible complex, I mean a single system which is composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts, the system to effectively cease functioning. An irreducible complex system cannot be produced directly by slight, successive modifications or precursor systems, because any precursor to an irreducible complex system that is missing a part is by definition non-functional. So Professor Behe admitted in reply to my critics that there was a defect in his views of irreducible complexity, because while it purports to be a challenge to natural selection, it does not actually address the task facing natural selection. Professor Behe specifically explained, quote, the current definition puts focus on removing a part from an already functioning system, but the difficult task facing Darwinian evolution, however, would not be to remove parts from a sophisticated pre-existing system. It will be to bring that system components to make a new system in the first place, end quote. In that article, Professor Behe wrote that he hoped to repair his defect in future works. However, he has failed to do so even 4 years after his defect. In addition to Professor Behe's admitted failure to properly address the very phenomenon that irreducible complexity purports to place at issue in natural selection, Drs. Miller and Patron testified that Professor Behe's concept of irreducible complexity depends on ignoring ways in which evolution is known to occur. Although Professor Behe is adamant in his definition of irreducible.
1 minute, David, 1 minute.
He says that a precursor is missing parts is by definition non-functional. What he obviously means is that it will not function in the same way the system functions when all the parts are present. For example, in the case of the bacteria flagellum, removal of a part may prevent it from acting as a rotary motor. However, Professor Behe excludes by definition the possibility that a precursor to the bacterial flagellum functioned as a rotary motor, but in some other way, for example, as a secretory system. As an expert testimony revealed, the qualification of what is meant by irreducible complexity renders it meaningless as a criticism of evolution. In fact, the theory of evolution proffers expectations as well-recognized, well-documented explanations for how systems with multiple parts could have evolved through natural means. Expectations means that some precursor of the system.
10 seconds.
Thank you. So, by defining irreducible complexity in the way that he has, Professor Behe attempted to exclude the phenomenon of expectation by definition fiat, ignoring, as he does, abundant evidence which refutes his argument, and all of that would also refute Otangelo, and we will have a lot more fun going into this from in the open discussion. So let's do the open discussion, and I'm actually reading from some of the highlights from the actual Dover decision. So this is actually what the judge wrote in handing down his decision.
Okay.
So,
okay, awesome. Well, just on time as well. So we're done the openings. We are done the rebuttal rounds. We are now going into a discussion. We'll try and keep it as equally timed, as cordial and respectful as possible. I'll do moderating as needed. Since David just ended with his rebuttal, as always, what we will do is we'll hand it over to Otangelo. If you want to make a couple points in the form of a rebuttal or ask David a question, and then we'll kind of go from there. Otangelo, so the floor is yours to start us off in this discussion. Go ahead.
Yes, I would like to share the screen again, and I would say some things and address them about what David said in his reply. Can you guys see my shared screen?
yes.
Okay. First of all, in regards of the Dover trial, that is something that is still being mentioned 15 years after it happened. And my reply to this is ask any real scientist if he thinks that the best way to proceed in scientific truth is to have courts of law decide what scientific theories should be accepted. Then to his second point in regards of irreducible complexity, and we can come to this in our free discussion afterwards, my reply is natural selection would not select for components of a complex system that would be useful only in the completion of that much larger system. I will explain what I mean by that afterwards. A minimal amount of instructional complex information is required for a gene to produce useful proteins. A minimal size of a protein is necessary for it to be functional. Thus, before a region of DNA contains the requisite information to make useful proteins, natural selection would not select for a positive trait and play no role in guiding its evolution. Then the third question which you brought up, David, in regards of if intelligent design is science, we have to make a distinction between operational and historical sciences. Operational science addresses how things actually operate and work. And when a biologist sees a protein which he has no understanding of its function, then he will try to find out what does that protein do, what is its function. But an entirely different question is, how did that protein originate? How did it come to be? And that is a question which is addressed by historical sciences. And historical sciences, they cannot be empirical in the sense you cannot prove, for example, if protein X emerged by evolution or by intelligent design. All we can do is observe the protein and then make informed inferences in regards of its origin. So there is a philosophical circle around historical sciences, and that is what is being debated. Abiogenesis and macroevolution, or the origin of biodiversity by evolutionary means, that cannot be proven either. It is far too much time back that we could go back there and see what actually happened. So that has also nothing to do with empirical science. So I would say that both propositions, macroevolution, abiogenesis, they are not grounded in empirical science, and therefore they are not in the category of operational science, where they can be made predictions and so forth. And it stays in the same ground as intelligent design. What we can do is just apply abductive reasoning to the best explanation and observe what happens in the natural world, and then make past predictions to what could have happened in the past to explain their origins.
Okay, perfect. David, make sure you unmute yourself. Of course, we keep it equally timed. Go ahead, respond to whatever you'd like from Otangelo, and we'll keep it going from here. Go ahead.
Yeah, well, thank you. Well, first of all, this is a debate on evolution versus intelligent design. This is not a debate on abiogenesis, although I would love to have that debate with anyone at a future date. I would rather stick to evolution. Evolution is a theory on the biodiversity of life. It is not a theory on the origins of life. These are addressing two separate questions. Evolution addresses what happens after life got here, and abiogenesis, or the origins of life, is a completely different question. It is perfectly possible that God exists and evolution is simply the process by which he created. It's very possible that there, in my opinion, there's no contradiction between God and evolution. Next, you brought up the two types of science, the historical and the operational science, which is a favorite from Ken Ham and the Pseudoscience Mill Answers in Genesis. This, of course, is completely incorrect. So the two types of the historical and operational science is just, this is what we call not even wrong. If we were to examine a murder scene or whatever, what we call that, oh, he's being the historical science guy by trying to figure out who did it. I figured out what did it. The observation science is, okay, there's the body and all that, but now the historical science is something else. No, that's simply not how it works. It's simply not even wrong. And then let's see, what else did you bring up? Yes, you brought up the Dover trial. the reason why I brought that up is because was a very important trial, and Dr. Behe and I actually brought up the actual reasons behind why the judge actually brought down that case. Let's actually look a little bit more into the reasoning of the Dover trial case. So one of the quite interesting things that Dr. Behe actually brought up was the immune system, and the judge quoted in one of his notes from the trial is the immune system is the third system which Professor Behe had applied the definition of irreducible complexity. Although in Darwin's Black Box, Professor Behe wrote that not only were there no natural explanations for the immune system at that time, but that natural explanations were impossible regarding its origins. However, Dr. Miller presented peer-reviewed studies refuting Dr. Behe's claims that the immune system was irreducibly complex. Between 1996 and 2002, various studies confirmed each element of the evolutionary hypothesis explaining the origins of the immune system. And then I'm going to repeat that last sentence. Between 1996 and 2002, various studies confirmed each element of the evolutionary hypothesis explaining the origins of the immune system. And in fact, on cross-examination, Professor Behe himself was questioned concerning his 1996 claim that science would never find an evolutionary explanation for the immune system. He was presented with 58 peer-reviewed publications, 9 books, and several immunology textbooks and chapters about the evolution of the immune system. However, he simply insisted that it was not sufficient evidence for evolution and simply was not good enough. In fact, in other words, he kept moving the goalpost. So the question that I want to kind of go into now is, you would obviously agree that speciation happens, correct?
Yes.
Okay, sure. So what I kind of want to do is start forward and kind of go backwards and see where exactly the extent of common ancestry breaks down. So for example, let me ask you a question. Do you think that the chimpanzees and bonobos share a common ancestor?
Oh, I don't know. I have not studied that.
Okay, sure. So let me ask you in this way. How would we know if two organisms share a common ancestor or if they don't?
I don't know that either.
Okay, that is quite problematic from your perspective, because I've shown through the genetic analysis how we figured out how cats diverged and evolved over time, and when and where they speciated and diversified, and we can do the same thing with humans. Would you at least accept that the cats, the paper that I showed you with cats, would you at least accept the basics of the paper that they have a common ancestor and diverted like that?
Yes.
Okay, sure. So when we apply that same principle with, say, humans, chimps, and bonobos and our close relatives, would you then say, would you accept that as well? Because using the same DNA molecular analysis, chimps and humans share a last common ancestor about 7 million years ago.
Well, I think you should debate that with SFT because he is a specialist in that. I've not studied that topic, so I'm not the right person to debate with you human origins.
Okay. Well, just one sec, David. No, I just want to make sure that we're doing as equally timed as possible. Maybe if Otangelo wanted to address a few things and feel like he was getting enough time as well, and then maybe also proceed to ask a question, and then we can go back to David to ask a question.
Okay, so David, he tries to refute irreducible complexity. He brought up the immune system. Of course, science since 2005 has advanced a lot. And I could actually read a text of amazing evidence how the immune system demonstrates design. But since in my second talk, I did read stuff, so I would like not to do that again. But I would like to make an analogy and then an exemplification to illustrate why I think that irreducible complexity in biology is very well established in a broad range of biochemical systems. It's not just the very well-known flagellum or the immune system, that these few systems which have been brought up by Behe and which are commonly being used as an example. And I would like to give you an example, and as an analogy, I would like that you imagine a photovoltaic system. a photovoltaic system uses sunlight to capture energy from the sun and then transforms it into energy which is stored in batteries. And that photovoltaic system is made of 5 distinct parts which operate in an interdependent system and manner together. And if one of these 5 parts is missing, then the system is unable to store energy in the batteries. And the first part of these 5 parts are the solar panels. If you don't have the solar panels to capture the photons, the light, then you cannot transfer that in the subsequent process and pathway to then in the end store energy in the batteries. Now, in order to make these solar panels, you need complex factories, and nobody in its sane mind would make only the solar panels without visualizing the whole photovoltaic system with all 5 parts which are necessary in order to have in the end the electric energy stored in the batteries. So you need to have foresight to see whatever you need, and then you need to make the factories for each of these single parts in order to in the end that then to have that photovoltaic system. That means that in order to have these sensitive cells, the solar cells, you need a complex factory. Now, if you start at the solar panels, there is nothing done. The system is irreducible. It is complete. And we have here an analogy to photosynthesis, to oxygenic photosynthesis. What happens? Chlorophyll is the molecule which is, as an analogy, the solar panels. It captures the light, then it funnels the ATP synthase to make energy, and that energy is used to drive Rubisco, which is the molecule which transforms carbon dioxide in glucose. And that is a system made of 25 very complex protein systems which have to work all together. And if even just one protein like Rubisco or ATP synthase or photosystem 1 is missing, you have not the product in the end, which is glucose. Now, the biosynthesis pathway to make just chlorophyll is extremely complex. It requires 17 proteins, which work like in a production line assembly process together, where there is the product handed over to the first protein, then to the second, to the third, until to the 17th protein, where in the end you have the chlorophyll molecule produced. Now, all these proteins in that production line system would have no use if they would make the chlorophyll molecule and only the chlorophyll molecule, because the chlorophyll molecule by itself has no use. It has only use if it is embedded in the antenna complex, which then funnels the energy to the special chlorophyll pair, which then transfers the electrons to the ATP synthase, and then fuels the proton important and then the ATP synthase makes the ATPs, which then follow the rest of the process. So what do I want to say with that? I want to say that this is an irreducible complex system where all proteins and parts have to work together, and chlorophyll by itself, produced by a complex biosynthesis pathway, would have no use. Okay, so I don't know if my explanation was confused or so, but I hope that with the analogy, I was able to bring through why I think that this is an irreducible complex system.
Awesome. Thank you, Otangelo. David, as always, we get equal time. You can respond to whatever you'd like from what Otangelo said, and then you can also follow up with a question if you'd like to. So go ahead, gentlemen. Go ahead, David.
Yeah, definitely. How much time do I have? How much time was that standing?
It was roughly 3 minutes, but take your time, whatever you feel like you want to address, and you can also follow it up with a question if you'd like, and keep it free flowing. Go ahead.
All right, yeah, definitely. Let me see. I'll see if I can pull up my screen real quick in a few minutes. Maybe I'll do that next. So yeah, again, I'll just start with a basic question. I brought, go back a little bit even further. The analogy between the machines and the life, I believe, is a false analogy. It's a false equivalence because you're not comparing like with like. organisms and biochemical structures have the ability to reproduce and pass down modified offspring, whereas mousetraps and others type of stuff like that simply do not. So you're not comparing like with like. That's one.
Okay, can I answer to that, David?
Sure, when I'm finished speaking.
Okay.
So yeah, so you're not comparing like with like, but again, but again, there's many evolutionary mechanisms for which in irreducibly complex parts kind of involved. Now, let me ask you a question, Otangelo. I brought up the issue.
Before you ask, David, I would like to reply to your point that you made that life reproduces while human-made machines do not. Okay. Now,
Okay, sure.
also DNA replication, which is what you are addressing now, is an irreducible complex system. In prokaryotes, we have 30 proteins which have to work in a joint venture together in order to have DNA replication. And if you have just helicase, for example, missing, which had to emerge prebiotically before life began, then you have no DNA replication. And in prokaryotes, we have about 30 proteins, and in human DNA replication, up to 600 proteins, which all have to be there in order to have DNA replication in the cells of humans. So that is basically a self-refuting argument, David, because DNA replication and self-replication is actually the epitome of manufacturing. We are far from being able to implement that kind of complex technology. We have tried. There is the Neumann machine, which was invented by a scientist which name is von Neumann in the 40s. And it has stopped there because it is very, very complex. To have autonomous self-replication is the epitome of manufacturing, and that is far from being achieved by human invention.
Okay, let's see here. Let me go back. So I brought up the issue of HIV and the evolutionary origins of HIV from SIV. Let me start by asking, would you agree with the basic model that HIV was modified, evolved from SIV?
I've not studied that.
Okay, would you at least agree that HIV is irreducible complex?
I've not studied that.
Okay, so what is your whole case for intelligent design is irreducible complexity? What would you consider to be irreducible complex? HIV seems to be irreducible complex to me, even though we know exactly how it evolved and the exact mutation steps that was necessary for it to be able to infect humans from the SIV ancestor.
Well, as I said, I've not studied your case, so I cannot give you an answer to that.
Okay, so what would you consider something that is not irreducible complex? Can you give me an example?
That's a good question. I think that I don't know.
Okay. So I need to have some type of groundwork or framework for where I can actually argue with. Because if you don't even know what's not irreducible complex, then I don't know how I can.
Well, I would say there are discrete things which cannot. It depends where you set the parameter. Like, for example, DNA is composed of 3 parts: the phosphate, the ribose, and the base. It is already an irreducible system, because if you want to have that molecule to be an information-bearing system, then it requires the 3 parts.
All right, let me share my screen real quick. Chrome tab. I think I kind of really want to hammer in this point just to see what we're dealing with. Can you see this quite all right? I kind of want to kind of lead into this issue kind of with universal common ancestry, because this is where our main disagreement is. We are eukaryotes, so I'm just going to zone in on the eukaryotic organism. By the way, I just remembered that you brought up the issue of how single cell became multi-cell, correct?
Mm-hmm.
Okay. Before I bring up that, we actually have observed firsthand a de novo origins of multicellular organisms. So what actually happened in this experiment was a group of unicellular organisms actually evolved to become multicellular in response to a selective pressure, which is in this case predation. So in this case, we actually do have reasons to believe that multicellular is not that hard to actually get, and this happened in a pretty short experiment. The transition from the unicellular to multicellular life was one of the few major events in the history of life that created new opportunities for more complex biological systems. So what happened was they subjected the populations of the unicellular green algae to selections. They put a selective pressure, which is one of the driving forces of evolution, by the filter feeding predator, I'm probably going to butcher this Latin, the Parmencium tetraola. Two out of the 5 experimental populations actually evolved multicellular structures that was not observed in the unselected control populations within only 750 asexual generations. So it's not that much of a stretch. This is irreducible complex. The multicellular is the irreducible complex, I guess you would consider. And yet here we go. We actually observed this firsthand from going unicellular to multicellular.
Well, it is possible that Chlamydomonas, which are actually amazing green algae, if they already had proteins like signaling proteins or cell adhesion proteins, even if they were unicellulars, already prepared to be able to make multicellular filaments. But that doesn't mean that there was an evolution event there. Actually, you need both, that is cell signaling and cell adhesion. You need these two mechanisms at once. If you have only cell signaling, but you do not have the ability of a cell to adhere to another cell, then you cannot have multicellular cellularity. And the opposite is the same case. If you have the ability, if you have cell junctions where a cell adheres to another cell, but you have not the ability for these two cells to communicate together, then you cannot have the multicellularity. So this is also irreducible. You need to have both functions at once.
Okay, so I seem to think that you're kind of shifting the goalposts a little bit. What I really need from you, just to understand, and let me address your point real quick. The multicellular structures that were evolved into the selected populations, the multicellular structures were not observed in any of the control populations. So again, this is completely a de novo evolution, new traits that evolved into a multicellular organism. So again, what I really need just to be able to start addressing this is a criteria for what can be irreducible complex. And once I show you this, you're kind of shifting it down a little bit more. So again,
well, the question is, what are we saying that cannot be reduced? First, you need to set a goal. For example, if you have a car, then the thing is, what can be reduced in order for the minimal functions to be preserved? And then you have the wheel, you have the engine, the several parts. And if you remove the engine, then the car cannot work anymore. So the car is irreducibly complex. And the same goes with any other things in biology, like ATP synthase, which is a nanoturbine, which is made of 7-8 main parts. If you remove one, then the nanoturbine cannot work anymore. So it is irreducibly complex.
All right. Sandy, can you share my screen again? Again, I'm just trying to understand your position a little bit more. So again, we're back here in eukaryote. We have 3 major clades of eukaryote. We have Biconta, which Pseudoflagella, any other eukaryote organisms in the Biconta. Then we have Excapata, which is a major subgroup of the unicellular belonging to the eukaryote. And then we are Unicoda, which is one pole. So again, is it possible that we have a eukaryotic organism and that this organism eventually diverges into these various populations. Is this possible at all?
Yeah.
Can these clades be related?
Yeah, it can be. It is possible.
Okay, sure. So now let me go into this clade here. We have these two clades, the amoebas here and the animals. So again, we have this going here and here. Is that possible for it to diverge further?
Yeah, I would have to go into the biochemistry and see what the differences are in order to give you an informed answer to that.
Okay, that's a fair answer. So going in here, this clade is extinct. So we have Brevetidae and these groups. I mean, is it possible that they could diverge further? So the question that I'm asking is, where exactly does the extent of common ancestry stop?
That is a difficult question to answer, because all this classification of phyla and kinds and so forth, that's very complex. So I don't think it makes sense to go into that. It is a difficult question, David.
Yeah, I understand it is a difficult question, but the whole debate, the whole purpose is intelligent design and evolution.
Well, I can give you, David, I can give you a formidable example of what would be irreducible. For example, there is a threshold of the first last universal common ancestor in regards of its complexity. And if you go lower than that threshold, then that organism cannot be alive.
Okay.
So there is a threshold between what is alive and what is not alive. And we have an example of this. Like, for example, Pelagibacter ubique is a bacteria which is one of the smallest bacteria known, which is free living, and it has about 1.3 million nucleotides. And if you go lower than that, then you go eventually to a mycoplasma, which is a parasite which lives in the human body, but it doesn't synthesize all the building blocks which it requires to be free living. And if you want to have an example of a minimal complex free living cell, then Pelagibacter ubique, which I said before, would be an example. And that is irreducible. If you remove just one of the 1,300 proteins which it has, then it cannot be kept alive. If it doesn't have ATP synthase, it cannot produce energy, it cannot be alive. If it doesn't have helicase, it cannot make DNA replication, it cannot be alive and perpetuate. If it doesn't have topoisomerase, then it cannot disentangle the DNA strands, and then it cannot it cannot make daughter cells, and it cannot perpetuate life. And I can go on and on. So there are these 1,300 proteins, they are irreducible. If you just remove one of those, you cannot have life. That is a formidable example of irreducible complexity.
So basically what you're saying is that any form of life pretty much is irreducible complex from the start?
Well, the concept of irreducible complexity is that you require to be able to maintain the basic functions of a system. And of course, if you have a Rolls-Royce is a car, but a Volkswagen is a car as well. Of course, you can reduce a Rolls-Royce to a Volkswagen and have a simpler car, but the Volkswagen will still maintain the basic function of the car, so you can reduce it, but there is a threshold where you cannot reduce it further.
Okay, sure. So I'm trying to steelman your understanding a little bit as to like what actually, in your opinion, would actually refute irreducible complexity, because I gave you examples of an irreducible complex feature that we know evolved, and you just said you when to study more, which is a perfectly valid answer.
Yeah, I've not studied HIV, so I cannot give an answer to that.
Okay, sure. So we know that certain bacteria and I think E. coli evolved several new functions. I think also through various mutations evolving the ability to digest nylon. Would that be irreducible complex? They have like various mutations that have to occur in order to do that. Would that be?
Well, I think that all organisms, David, they have a pre-programmed ability to adapt to the environment and to mutate in a limited manner in order to be able to survive in different environments. But
okay.
that is not a function that is evolving. That is a function which is already pre-programmed right when life started. If you do not have that ability right from the beginning when life started, then you cannot have life, because life has to have the ability to be able to adapt to the environment.
Okay, so you said there within a limit. What is the limit? Do you know what the limit is?
I gave an example with the limit of Pelagibacter, which would be basically one of the smallest possible bacteria. And if you go lower, if you take away one of the life essential proteins, then you do not have life anymore.
Okay, trying to think of the best way to approach this. Let's see here. I'm just trying to think of a way to really go through and attack this. So we know that there's like various different mechanisms and different pathways for which new structures and new proteins and all that can and do evolve. So
do we know that how?
Because they've been observed, such as what I already showed you with HIV. They and also with this nylon, and in fact, we also hold on a second. Let me finish. Let me finish real quick. Mm-hmm. We also, very interesting, there is at least one species of snake that is currently is birthed to both live and eggs. So we can actually see the process of evolving a placenta in real time and evolving the ability to give live birth. So again, we have seen this happen in real time.
Yeah, I mean, maybe that is speciation, maybe it is adaptation, maybe it is microevolution. I think that is very well possible. What I said in previously is that what is in dispute is primary speciation, where new limbs arise and eyes and ears and different body structures and architecture, big changes. This is what is in dispute.
Okay, sure. So we both agree would you agree that speciation is macroevolution?
Secondary speciation is macroevolution, yes.
Okay, what exactly do you define as secondary macroevolution?
Okay, secondary speciation is when populations separate and then they cannot interbreed anymore. Then you have two different populations. That is a speciation of second degree, but what has not changed is the organismal structure of these populations. They remain basically similar, but first or primary speciation is when big changes occur, when, for example, the transition from fish to land, or becoming whales, or the arising of eyes or ears, that kind of differentiation, which is big changes.
Okay, sure. So we know that speciation and all these changes evolve slowly and gradually. So what happens is the accumulation of all these mutations and these quote unquote secondary speciation is evolved as the effect is effectively over time brings in these primary macroevolution and primary speciation thing that you were talking about. Now, remember that according to the law of Mendelian basic evolutionary development, you are always part of the same clade that your ancestor belonged to. One kind of organism simply never changes into a new kind of organism. And these are actually really good points that you brought up. Can I share my screen again? Because I really like that point you brought up about evolution. Can I need to share my screen. There we go. We do know basically and we do have transitional fossils of this as well, of how these quote-unquote macroevolutionary structures evolved. Multiple regulatory modes, for example, are required for scale-to-feather conversion. And we have the genes. We know exactly how they do that. We identified feather-associated genes using genomic analysis. The candidate genes were tested by expressing them in chickens and alligator scales forming regions. The ectopic expression of these genes induced intermediate morphotypes between scales and feathers, which revealed several major morphogenetic events along this path. Localized growth zones, formations, the follicle, and and et cetera, et cetera. So we do by studying the genome, we have a very good idea of how they evolved. We have the fossil records for them. We know many types of dinosaurs did in fact have feathers. Let's see here, what else? Human hair bird. This is another good. Here we go. Human hair, bird, feathers all came from the reptile scale. This study could end a long-handed debate in the evolutionary biology. So again, this is all by looking at the genomic evolution and all that. Let's see here. Here we go. Here's another good paper, Evolutionary of the Heart. Mainly, yeah, that was not the right paper. Here we go. Okay. I lost where I was at, but yeah, so the point of the matter is we do see these. We know roughly from both the fossil record and genetics about how these quote unquote primary macro things, primary speciation happens. With that, I'll let you respond to that.
Well, first of all, before I could give an informed response to you, David, I would have to read the paper and go into the molecular level and then see what mechanisms that they propose and then see if that what they claim is sound. Okay. Now, what I have learned so far is that big changes, they do not depend only of genetic variation, but in special also on epigenetic mechanisms and languages. And I have categorized almost 30 different epigenetic codes and languages, which are all involved in a systems level in constructing organismal form and structure. And that starts already in the construction of the variation and differentiation of each single cell. Like, for example, a neuron has a completely different form and structure and function than a blood cell, but they have the same genomic information. So, of course, you need to ask what generates that differentiation starting from the single cells. The human body has about 200 different cells. And then how do you get to the organismal complexity and the structure and the tissues and the layers and so forth? And it is not just genetic information. It is much more complex than that. It goes even into photonic information signaling by light. And I have a video on that. If you want, I can share here on the screen all the mechanisms which I have collected, which are actually involved in organismal construction.
Okay, sure. But before you share that, I wanted to give a little bit of a chance to respond. I totally respect the fact that you want to take a little bit more time to research into the papers. I know I said that to Standing for Truth in many of my debates, and he has always been very respectful in accepting that. So I will give you the same respect he has given me. I will. If you want, I can send you the papers and you can look at them if you would like.
Sure.
Okay, and a couple more examples that I want to show you. Chrome tab, key events. Can I share my screen again? Here we go. Key genetic events underlining the fin to limb evolution. Here we have the study in cat sharks to reveal how the alteration in the expression functions of certain genes in limb busts actually underlie the evolution of fish fins to limbs. Alteration in the expression of the findings are reported here. Again, here are we do have the genetic possibilities. Let's see here. I do want to bring up some of the really interesting transitional fossils as well, because that I think is pretty solid evidence that goes along with the genetic evidence. So while I'm getting that set up, feel free to take your time to respond.
Okay, go ahead.
I just wanted to give you some time to respond to what I said while I was looking up
Well,
the.
I honestly, David, I think that comparing fossils is completely meaningless. As Behe said, that the action or the mechanisms which define organismal form is on a molecular and biochemistry level. And there is where you have to see the real mechanisms which are defining organismal form. And unless you do that, then it is completely pointless to try to compare fossils one to another and say, oh, we see here similarity. That means it has to be a process of evolution. This is, in my understanding, completely meaningless.
Okay, sure. So I will be responding that you're correct that species and all is definitely determined more in the molecular level than in the morphological level. So it is definitely very difficult when looking at fossils to see which organisms are of the same species and which are not. I grant you that 100%. So there's definitely a lot of guesswork going into it by mostly looking at morphology and where they are in the fossil record and what layer they appear in. I would also point out that creationists actually do the same thing. They have the same hypothetical framework by trying to figure out what kind, what Barron particular dinosaur fossils actually belong to. So it is possible to get a guess, but again, without the actual DNA, you're correct. It is definitely difficult, but we can definitely see hypothetical for that. Hypothetical based on mythology.
I can give you an example, David, why I think that the genetic information by far does not suffice to explain multicellular complexity. For example, our genome, the human genome, has about 3 billion base pairs, but we have 37 trillion cells. Now, each cell has to be specified in regards of, I think, 19 different issues, which is the form which it has to have, what kind of cell that it will become, the size that it has to have, how many replication events that it can have and then self-destroys, and so forth. And all this information, and it's special, where does it locate in the body? And if you have 37 trillion cells and each has to be specified where in the body that it has to be, then of course, if you have only 3 billion nucleotides and the genome, it is not sufficient to have all the information for that. And for that reason, I say that the information to construct multicellular organisms is not only genetic, it is also epigenetic. And for example, the glycan language or code on the surface of membranes of the cell, it is far, far more complex than the genetic information. And it also is involved in the specification of location and communication with neighboring cells and so forth. So all this information has to be taken into account in order to explain organismal form. And science is just in the beginning to unravel that kind of complexity. We almost have no explanation of the code of all these epigenetic languages. We don't even know where this information is stored. We have amongst almost 30 different languages, just about 2 or 3 languages which we know how they are stored. Like in the histone tails, there is information, and like I said, the glycan code, we know the structure of the glycans, of the glycoproteins. That structure also bears information. But there are many other codes which we do not know how that information is stored. So we are still in the dark in regards of many fields to explain organismal complexity.
Okay, sure. Hey, Standing, how are we doing on time? I'm just kind of curious to see how much further we have before we go into Q&A.
We're doing good. I've got 10 more minutes on the clock for the discussion and then Q&A, if that works for you, gentlemen.
Yeah, that sounds good. I'm really enjoying this debate. How about you?
You know, this has been a great debate. We got over 40 people in the chat. We got a lively chat. There looks like there's some side debates going on in the chat. You guys are bringing the numbers. It's a lot of fun, engaging. So what I'll do is I'll give since I've got 10 minutes left on the clock, let's do 7 more minutes. And the last few minutes, I'll let you guys wrap it up in the form of a concluding statement. And then we'll just jump right into questions.
That sounds good. And just before I get into there, all the keyboard warriors in the side chat, get your butts in here and actually debate like a man. That's what I say, at least.
I would agree with that, David. I would agree with that. You, and that's why we appreciate you always being willing to come on and debate, because these discussions are important. So anybody in the chat, anybody that follows our channel knows that we host weekly debate, sometimes multiple a week. So anybody interested in live debate, let me know and we will set it up. So anyways, I'll hand it over to you guys to finish it off for us. Keep up the good work. It's been a great debate.
Okay, sure. So I kind of lost my train of thought. I was going to bring up some of the transitional fossils as well, because I think the molecular data, as well as the fossil record showing these gradual changes over time, actually does a great deal of shows a great deal of evidence for evolution. Can I share my screen one more time? Transitional fossils. I think my personal favorite is the Tiktaalik fossil, and one of the reasons why I love the Tiktaalik fossil so much is that they actually predicted where exactly they would actually find this fossil before they actually found it. They said, okay, we're going to take the basic evolutionary framework and what we know about the time scale and say, okay, if the fish to land animals is true, we should find a structure for we should find a intermediate fossil that looks somewhat like this, and we should find it in this layer, and this is exactly what they found. And there is good evidence that the Tiktaalik actually had both lungs and could actually had lungs and lungs and gills, and many others as well. Probably the representative group from which tetrapods evolved. And we see dinosaur to birds, very well documented in the fossil record. The Acrinodus, which is found that probably depredates even the famous Archaeopteryx. The requirements, feathers, and all. And so these transitional fossils do exist, and they confirm what we see in the molecular data as well. And it conforms exactly to what we would see. So there's no problem with the primary speciation and stuff like that. So I'll let you respond.
David, as I told you, for me, what is relevant is looking into biochemistry, into molecules, and there I find the real mechanisms which are involved in organismal construction. And it's not genes alone, it is also epigenetic information. And so that is, in my understanding, the reason why the evolutionary mechanisms are inadequate to explain organismal form. We have, and actually even there are proposals like the third way, for example, and they are including epigenetic information into the picture. So, but they exclude an intelligent designer. But science is going in that direction to acknowledge that epigenetic information and signaling networks are also involved in organismal construction. And then, of course, once these mechanisms are englobed into the picture and analyzed as well, we have to ask the very fundamental question, David, where does that information come from? And where does information transmission systems come from? And you have there several things that you require. You require the language. You require the information upon which you use the language to store that information. You need the information storage mechanism, the hardware, like DNA, which is a hardware like a hard disk. You need the information transmission systems that is from DNA to a messenger RNA, that is from DNA which is transcribed and then translated to the ribosome. When the protein is synthesized, you need other information to direct the protein to where it is actually employed in the cell. And you have vesicles which are tagged, and they know exactly at what location that the protein has to be directed to. You have tubulins on which molecular motors and taxis transport the molecules to the right destination. That is a molecular GPS system. You have to ask, where does that information come from? And to say, oh, that is all by a gradual evolutionary mechanism, slowly. No, you need that all at once. If you have all the information to make a protein, but you don't have the information where to bring it in order to be employed at the right place, you have nothing done. And I can go on and on. And that's why I say, in my understanding, is intelligent design absolutely required in order to have life and to have these ultra complex biochemical systems.
All right. Thank you, Otangelo. From my count, I think you probably brought up at least a dozen or 2 dozen of structures that you would say are irreducible complex. What I think would be actually beneficial, since I think we had a really good discussion, is maybe you spent some time in researching some of the points that I brought up, and we can have a second debate and maybe do just one or two at a time, because having 20 or so points that you think are irreducible complex are simply is not going to really happen in a 2-hour debate. So I would like to maybe see if we can discuss maybe one point at a time, maybe take a couple of these points that you say are irreducible complex, and maybe discuss like one at a time. When you want to do that? And maybe,
I can give you an example. You can investigate Mycoplasma mobile, which is a mycoplasma. It's a bacterium which has only 4 amino acid proteins, 4 proteins, and it uses just these 4 proteins for locomotion. And if you remove just one of these proteins, you do not have any locomotion anymore. These 4 proteins, they are not used in any other biosystem, only in that regard. So I can give you the link afterwards. There is a wonderful video, and you can try to figure out how you could reduce that system, and then you can give me an answer in an eventual second debate.
Okay, let me jump in here real quick. Did you guys want to now wrap up your points, unless you feel like you have wrapped up your points, and then we can move into our question and answer period?
Yeah, sure. I'm fine. I'll probably do like a 3 or 4 minute wrap up if you want. That'll be fantastic. And you want me or Otangelo to go first?
Yeah, that's a good question. If you'd like to, by debate standards, we could allow Otangelo to go first, since he started first in his opening, and then let you have the last word, David, if that works for you guys.
Yeah, that sounds good.
Okay. So let's allow Otangelo, take a few minutes, take your time, no rush, wrap up the discussion, address any points you feel you want to address before we get into our question and answer. Then we'll hand it over to David to have his last word. Go ahead, Otangelo.
Okay, I believe that the big umbrella contributor to explain organismal complexity is pre-programmed instructional complex information encoded in various languages and communication through various signaling networks that act on a structural level, which are pre-instructed to respond to environmental cues, development, and nutrition demands. And they are apt to communicate, to crosstalk, to signal, to regulate, to govern, to control, to recruit, to interpret, to recognize, to orchestrate, to elaborate strategies, guide, and so forth. All codes, blueprints, and languages are inventions by intelligence. And therefore, the genetic and epigenetic codes and signaling networks and the instructions to build cells and complex biological organisms were most likely created by an intelligent agency. Okay, that's my final statement.
Awesome. Well, thank you for that concluding statement, Otangelo. I want to say great debate, gentlemen. Great discussion, really engaging. The chat was awesome, really lively, as usual. So, David, we're going to hand it over to you for your concluding statement as well. Go ahead.
Well, thank you, Standing for Truth, for having me back on your channel. It was great being back, and thank you to Otangelo for this fruitful and lively discussion. I really enjoyed our chat tonight. So I just kind of wanted to group my arguments together. I believe that evolution is the best model that we're looking at. Nothing in biology makes sense except in the light of evolution, and that evolution does make these testable predictions. And not only is it testable predictions that turn out to be correct, but it's very useful for the in the field of medicine and learning about the origins of new viruses. Just a little bit of a recap. I think that despite me being at a right now, I am going on my 17th hour of being awake, so I am absolutely exhausted. But I think I did pretty well despite my lack of sleep. Just kind of a couple of points. I asked Otangelo several questions that he did not know, and that is perfectly fine. And I totally respect the fact that he said, I don't know. I asked if he would consider HIV to be irreducible complex. He said, I don't know. I asked him whether or not too closely, how we could tell if two organisms were related together. And he said, I don't know. These are serious problems with his position. Even I asked him a pretty easy question, whether or not the chimpanzee and the bonobo were closely related. Again, he said, "I don't know," and these are valid answers. And but I do want the audience who are taking notes to remember that. Couple points. I think that I would definitely want to do another discussion with you where we discuss some of the irreducible complexity parts. And so, yeah, I am looking forward to our next discussion, and I'm definitely looking forward to being back on this channel again. And that concludes my statements.
Awesome. Well, thank you for that concluding statement once again. Great debate, gentlemen. We have a good question and answer as well. We'll keep it roughly because I know it's been a long day for you, David. Thanks for sticking it out. Thanks for having a coffee just for us. And given the debate your full energy. So I like that. Let's get right into the questions then. I am going to, let me see, I saved them all in here. And let's start with number one. Before we gave our openings, I think I went over the 5 super chats we had at the beginning. So thank you, everybody. Also, reminder, Salvador Cordova is having an after show after this debate. So make sure you stop by the after show. Okay, so let's get into the first question. First question from CIH. Question for David. David, you're kind of in the lion's den because most of these questions are for you. So
I figured.
we appreciate you being a good sport. You're on a creationist channel, so it's what is to be expected, I guess. So here we go. So what we'll do then is when you give a response, we can allow Otangelo to give a brief response, and then we will just allow the person who the question is for to have the last word. Okay, so question. How can evolution account for the second law of thermodynamics? David, question is for you. Go ahead.
It doesn't have to. This is what we would consider not even wrong, and I am working on some stuff about the second law of thermodynamics. We do know that complex stuff does happen in nature all the time. Earth is an open system. It is not a closed system. And even in space, we know that stars, planets, and galaxies all form naturally. We've seen the emergence of new stars, highly ordered structures. So second law of thermodynamics is not evidence against evolution.
Okay, thank you for that response there, David. Did you want a quick response, Otangelo?
No.
Okay, let's move right on to the next one. So next question is, let me see. I'm guessing it's for you, David, and I'm right. There's a testable prediction. So, okay, so George Bond, $5 super chat. Thank you, brother George. Question for David. He says MO1 bacteria, 7 flagella, and 24 interconnecting gears to ensure tangling doesn't occur and maximizes power. How is this not irreducible complexity. Go ahead, David.
So that question is related to the bacterial flagellum, correct?
Yes, it looks like it.
Okay, sure. So this was actually dealt with in the Dover area school trial, and this is what actually happened. In the case of the bacteriophagogen, removal of one part may prevent it acting as a rotary motion. However, Professor Behe actually includes, by definition, the possibility that a precursor to the bacteriophagogen functioned not as a rotary motor, but in some other way. So that is possible as well.
Awesome. Well, thank you again, David. Otangelo, as always, I like to ask, would you like a quick response to that?
No, go on.
Okay, I appreciate that. So next question is from SFB Mob. Thanks for your question. He says, question for David. How do nucleotides form in a free-floating phosphate bath?
I am not sure what that question means, but that being said, we do know that nucleotides do appear in nature. So having the nucleotides, we already know the nucleotides do exist. So all what we really need is a catalyst for the nucleotides and all of the other stuff to come together. Not my field of expertise, but thank you for that question.
No problem. No problem. Thanks again for being a good sport. Thanks for the response. Otangelo, did you want to add anything to that question?
Yeah, of course. Nucleotides, they are not free floating in nature. They are very complex molecules, and science has absolutely no clue how RNAs and in special DNA could have formed prebiotically. And that is a huge abiogenesis problem.
Can I respond to that real quick?
Yeah.
David, it's your question. You get the last word.
Yeah, sure. So I know we are definitely not anywhere near understanding the exact origins of life, though incredible research has been done. And recently, I think last year or the year before, they were able to get RNA bases through an experiment. So again, not a problem whatsoever. But thank you for that question.
Can I respond to that?
Well, I guess it was David's question. David, do you want Otangelo to have, if Otangelo does give a response, in all fairness and rules to the Q&A, David, you'd still get the last word, though.
Let's move on to the next question.
Okay. So let's see. Next question here. Actually, super chat just came in from Chris Keith. $5. Chris, thank you so much for the support, brother. So question is for, take a guess. It's for David. So David, what is the specific mechanism that allows for punctuated equilibrium? In brackets, he writes rapid change.
That is a fantastic question, and the answer is that is through a selective pressure. Evolution does happen slowly, but it can happen quite rapidly. If there is a change in the environment, and in cases of a mass extinction, organisms have to quickly adapt in order to adapt to these new climates. And so that is just one mechanism right off the top of my head that will allow for such drastic changes to happen so quickly. Right after mass extinction events is generally when we see a huge explosion in biodiversity. But
okay, thank you for that response there, David. Otangelo, would you like to add anything to that question?
Oh yeah, it's God who makes it. So that's my answer.
Awesome. Okay, well, let's move on to a question from LPP, logical, plausible, probable, in the house. He's coming at you, David. So here we go. Question for Neff. He says, all organisms don't require all 20 AA, but 99.999% of proteins do require all 20. So by AA, amino acids. So did the ability to synthesize all AA, amino acids, have to occur before evolution is viable?
I am not sure the answer to that question, but that's a great question and something that I will be researching.
No problem, Dave. You're doing great. Like I said, you're a good sport. You're here, and there's a lot of questions coming at you from our creationist audience, and not all of them are easy. So once again, I appreciate you being a good sport. Otangelo, did you want to have a response to that?
Well, the synthesis of amino acids is, of course, also a huge abiogenesis problem, which is unsolved. And in my understanding, there is no explanation, a naturalistic explanation for that.
Okay, well, we got a question. Actually, David, your question, so you can have the last word if you'd like.
Nope, I'm good.
Awesome. You guys are both very cordial and respectful. This was an awesome debate, very easy debate to moderate. So I want to thank you again. So question for Otangelo. From your best friend, Otangelo, speed of sound of gravity. He is here and he is enjoying. So he asks, how do you justify, Otangelo, that it needs design? It's stochastic, he says. Take your time, Otangelo.
It's not stochastic.
There you go. You're coming at speed and he's coming at you. So thank you for that response. David, this was a question for Otangelo. Would you like a quick response?
No, I don't have a response to that.
Okay, awesome. So question from Smoky Saint. Good question. The question's for both of you. So we'll have a little fun with this. Whoever wants to start. Question is, what, if anything, would change your worldview?
That is a great question, and I love asking that question. And I think that in the realm of debate, that is one of the most important questions you have to ask, and one of the most important questions you have to even answer. Because if your answer to that question is nothing, then you're obviously closed-minded, and that no discussion or debate is even going to be fruitful. So a lot of things that right off the top of my head. Finding a fossil where we shouldn't find it, such as a pre-Cambrian rabbit, would be quite detrimental to the theory of evolution. One of the creationists' absurdities, such as a bacteria giving birth to a human, would also pretty much falsify evolution on the spot. If we were to find a fossil that we shouldn't find, such as a crocoduck, that would also be pretty detrimental to the theory of evolution. Or if we find like a half tree, half human, that would also be pretty detrimental. If we were to do our DNA analysis and all, and we find that we find 99% of our DNA is shared with a banana, but only 1% with the chimpanzees and bonobos, that will be quite problematic on a genetic level as well. So thank you for that question.
Awesome. Thank you for that answer there, David. Otangelo, over to you for your answer. The question was, what, if anything, would change your worldview? Go ahead, Otangelo.
Well, it would not have to be just one thing. There are many issues that would be required in order for me to change my worldview. But in regards of science, of course, the origin of information systems, the origin of code, information, then information storage systems and information transmission systems. If that could be explained by natural means, that would be a big blow for intelligent design.
Thank you, Otangelo. That was a pretty good answer, I think.
Yes, great answer from the both of you. This has been a great Q&A. We're going on 2 hours, and we do have 2 questions, so we'll end it with these last 2 questions. The current question is on the screen from Jungle Jargon. He says, what exactly is the mechanism for writing instruction that is not there? At David Neff. So question for you.
I do not know what that question means. A follow-up question for him is, what do you mean by writing instruction? What does that even mean? I don't understand the question.
Awesome. Fair enough. Otangelo, maybe you understand the question a little better. I'm not sure. Did you want to have a response to that?
Yes, of course. Only intelligence is able to generate instructional information with specific goals. When you instruct something, then you want to achieve something, and non-intelligence is simply the wrong mechanism for that. It's only intelligence that can do that.
And David, it was your question. So as per the rules of the Q&A, you can have the last word.
No, I'm good.
Okay, so question here from Lena Powell. Thank you so much for the question. Lena Powell asks, David has mentioned nyelinase, but this is not a new function. How would you respond, David? Go ahead.
So the bacteria in question, I can't remember right off the top of my head. They were not able to digest nylon in the past, but they were able to digest nylon now due to being in a nylon-rich environment. And of course, nylon being a synthetic product, they simply could not have had the ability to digest nylon prior to the 1950s when it was invented. And I believe it was like a point mutation or an addition that was the cause of the ability to digest nylon. So thank you for that question.
Thank you for that answer, David. Otangelo, did you have a quick response there?
Yeah, the gene was already there. It wasn't expressed yet, but when the selection pressure came, then the bacteria was able to express the function. So that's not new information.
Thank you, Otangelo. And David, you can have the last word there.
Sure. So the obvious question is, what do you actually mean by new information? They obviously did not have it there. It wasn't there originally. And what would you consider new information anyway?
Well, I think it was simply not expressed. It was there. The gene was there, but it wasn't expressed. And when the selection pressure was there, then it was expressed. So it's not new information. It's not a new strand of genetic information which has developed de novo. It was already there, but not used yet.
Yeah, actually, I just looked it up. It was actually caused by a frame shift mutation. So, yeah, they definitely did not have that ability before. So, anyway, next question.
Okay, so we got a super chat from Caleb. Thank you so much, brother, for the support. You guys are awesome. Like I said, you guys are the reason why we are able to put in all the overtime to get you guys the content. So Caleb says, at Standing for Truth, for both, which requires more faith, evolution or creationism? What we can do, since the last question that was for both from Smokey, David, you started. So Otangelo, if you'd like to start with a response to that question, once again, which requires more faith, evolution or creation?
Well, I require very little faith in regards of intelligent design, and I require a huge amount of faith to believe that non-guided, non-intelligent mechanisms can bring forward what we see in the natural world, which is far more complex than anything that humans can devise with all their intelligence.
Well, thank you. Well, the faith is basically believing without evidence, and the problem is evolution is observed. So in that case, we don't have to have any faith for our position because it's observable, it's testable, it's repeatable. So there is no faith involved in accepting the demonstrable reality of evolution.
And last minute super chat, Doki Doki Bible Club, $5. Thank you so much, Doki Doki. Doki Doki says, tired David isn't enough. We want a bigger handicap next time. What do you say, David? See, we've got a fun chat. We keep this entertaining. The 2 hours has flown by. You guys have been great entertainment.
It's always a fun time.
Actually, last question for the night. SFB Mod, question for David. So one final question. We'll hit the 2 hour mark right on. So he says, why he claims Einstein, I'll put it up on the screen, why he claims Einstein was wrong that the universe is finite and closed.
So I guess he misunderstood what I was saying earlier about the second law of thermodynamics. Earth as a whole is an open system that receives energy from the sun. So the second law of thermodynamics does not necessarily apply to evolution here on Earth. And we know we can see, if that was the actual case, we should not be able to see like plant seeds becoming more complex and all. And even outside in the universe, yes, the universe probably is a closed system. But we still do see stars and planets forming even today. We know the process of star formation and planetary formation very well. Again, that also does not refute the second law of thermodynamics.
Okay, well, I appreciate you answering that question for us, that last minute question. Otangelo, would you like to say anything to that?
No.
Okay, so let's see. Yeah, it looks like we got them all. So guys, thank you so much for the super chats. Thank you so much for the super stickers. You guys are awesome. This was a great debate. 2 hours has flown by. I've really enjoyed myself, and I can assure you that the audience has as well. Guys, this Saturday, be here. Genesis Flood debate, Derek Barnes versus Nephilim Free. Derek Barnes, you will be familiar with. He was here just last week debating Ken Hovind. That was a really good debate. And I've also debated him myself. And guys, Salvador Cordova is having an after show right after this debate.
Yeah.
So make sure you go there, check it out. David, I imagine you're going to be there as well. He just put the link in the there we go. There's the link.
Okay, get there. I will probably be there for a few minutes. Probably won't be staying there the whole time. Is there a link to the StreamYard?
Yes. Yeah, we will. Actually, I don't think I have it yet unless Sal sent it to me, but I think Sal will probably do good by putting the link in the live chat for people to jump in, or we can send the link around through email to avoid any unwanted guests. But I want to give it to you guys for some final words, final thoughts before we close it down for the night. I want to thank you both again for giving us your time for this really good debate. We still got 40 people in the chat. People loved it. Great debate. So final word, gentlemen.
Yeah, just to make clarification, Doki's, there is not going to be an after show on my channel. I'll probably check into the after show and just collapse a few minutes after that because I am ready to collapse. There is no after show on my channel, so everyone go to Salvador. Go to Salvador's channel for the after show.
Awesome. And Otangelo, any final words before we close it down for the night?
Yes, thank you very much, Standing for Truth, for having me at your channel. It was fun. Thank you, David, as well, for this debate. You were very polite, very educated, very intelligent. I love to have this debate with you. And if you want, I look forward to have another one, and we can dig deeper in these questions and see if we can clear up some things.
Thank you. Yeah, that sounds great.
Amen. Amen. Both of you were good sports, and the debate was very cordial, respectful. These are my favorite kind of debates, where people are not talking over each other or monopolizing the conversation. This was just perfect, the perfect debate. So thank you so much, guys. Head over to Brother Sal's after show. Should be a lot of fun as usual. And Standing for Truth is out. God bless everybody. Good night.
Thank you.
For all those who appreciate the work that we're doing here on Standing for Truth,